translated and sort of by the machinery of bacteria. So sorry, for the physicists in the room that never thought about this, sort of basic introduction to biology. There are cells. Okay, but there are cells, and the cells have a DNA. In the DNA, there are instructions to build proteins. Okay, so what a cell has to do, it has to read the DNA, RNA, a messenger RNA, and the RNA is read by machines, read RNA and take amino acids and assemble proteins. Okay, so basically a cell, in order to produce the proteins that they need to do whatever they have to do, to make new cells, to grow, to survive, they have to read the instructions on the DNA and make proteins. What viruses do is that they inject their own DNA, and they force the cell to read that DNA instead of the cell DNA. And what the DNA has as an instruction are the instructions to make new viruses. So basically they enter, this DNA is read, and then there are many viruses that are sort of produced, and then at a certain point the cell explodes because there are many viruses, right? So the point is, virus enters, produce new virus, explode. Okay? There is also other things that virus can do. We'll talk about this later, but this is like sort of the system we have in place. And so this is bad for bacteria, right? Basically, it's a way in which you kill bacteria to produce new virus. Now you have a sensitive. You can imagine that you have sensitive bacteria, and then you have resistance. Okay? So you have bacteria that develop some mechanisms not to be infected by phages. Okay? And so the two kind of cells that we're going to look at are sensitive cell and resistant cells. And so the idea is that we start with sensitive cells, and then we want to study mutations that lead to resistant cells. And the question that we have is, do these mutations arise because there is the challenge, because there are viruses, or they arise independently? Okay? Is the setting clear? Questions about setting? Good. Okay, so this is the experiment that they did. So basically, you have this is a sort of simple, very simple biological, microbiological setting. So you have some bacterial culture, okay, to which you add phages, and then what you can do is that you can isolate phages. Okay, so basically, you have a way in which you can have a culture of phages. You have many phages in this culture. Okay. Then what you do is that you add bacteria to some medium, so to basic nutrients that are needed for life, some salts and some sugar, sorts of energy, and then you grow them, and you have a culture of bacteria. So basically, you have two big flasks where you have phages and where you have bacteria. Okay, and then what you do is that you take a petri dish, small dish, with some agar, so some solid medium, where bacteria can grow, and you put on top a nice layer of viruses. Okay? And then to this one, you add bacteria. Now, the way you add bacteria to this layer is that you don't like just pour your flask over. Okay? What you do is that you dilute the bacterial culture such that when you put it on the petri dish, there are very few cells. Okay? So the idea is, suppose that you don't have viruses. Okay? So you add your bacteria to a dish with only food. You dilute your bacterial culture so much that then on your plate there are only, I don't know, 50 cells. Okay? So there are only 50 cells, and so you have your plate where you add your bacteria very diluted a lot. Okay, and so when you dilute, when you start, there will be, let's say that these are not 50, they are 6. You have 1, 2, 3, 4, 5, 6. Okay, so basically, if you have 10 to the 9 cells and you dilute, then you expect to have on average 1 cell. Right? If if you have 10 to the 9 cells per milliliter, and you put this one, you dilute this one with water many, many times, right? You put one part of this in 10 to the 9 parts of water, you expect to have in 1 milliliter 1 cell, okay? So you do this such that you have, let's say, on average 10 cells. When you put this, you start from 10 cells that you cannot see, right? Because it's just 1 cell. You have 1 cell here, 1 cell there, etc. And then these cells grow, right? And when they grow, they form a colony that you can see with your naked eye. Okay? So then you inoculate this, then you wait, I don't know, 12 hours. I have no idea of this number. And then what you do is you do something very fun. Say that you wait 24 because you want to have a more work-life balance life. So you wait 24 hours, and then what you do, something very fun, you look at this and you count. Okay? So you count how many colonies and you say 6 colonies. Okay? And so this is actually a way to measure how many bacteria you have at the beginning. Right? Now, what you do here is that you can do it in presence of phages. Okay? If you do in presence of phages, the only bacteria that can grow are the bacteria that are resistant to phages. Okay? So if I started from if you start from your culture and you had, I don't know, 2 resistant cells and 3 bacteria, 3 non-resistant ones, when they grow, okay, you're gonna see just the colony of the resistant one. Okay? Is that clear? The setting? Any doubt about this?
Maybe somebody can explain that.
Can somebody do it? Can go downstairs? No. Okay. How do I? Okay. So, so the idea. Okay. So the idea is that the question that you can ask is, how many colonies do I expect at the end? Okay. So if I do this, and I count how many colonies of cells I have, in this way, I will count how many resistant cells there are. Okay? So the idea is to start with some bacteria that are sensitive to phages, and then count how many resistant cells I have at the end. Okay? Or better, I can count how many resistant cells I have over the total bacteria. Okay? So the question is, how many cells do we expect to count at the end? Okay? So let's start with the Lamarckian scenario. Let's start with the Lamarckian scenario. And so the idea is that what happens when the Lamarckian scenario is that we have our nutrient and we put, like, let's say, 1 cell. Now you never put exactly 1 cell, but you can imagine that you start with some small column. For the calculation, let's imagine that you put just 1 cell. Okay? You put 1 cell, and then this will grow, right, in absence of phages. Okay? So you go from 1 cell to n, let's say, 10 to the 9 per milliliter cells. Okay? And what happens in this process here is that what you can imagine is happening is that you go from 1 cell, and then these cells are growing exponentially over time. Right? So you have like a population that is growing exponentially over time. Okay? This is just a way to imagine that this is like a population expanding. Okay? So this population is growing exponentially. And then you arrive at the end, and you have n cells. Okay? And now what you do is that you take these cells here, and you put it on a petri dish with phages. Okay? So you have the challenge. So here you have challenge. Okay? And then you count how many cells are resisting. Okay? So at the end, after the challenge, only the resistant cell will survive. Okay? So let's say that we are in the Lamarckian scenario. Okay? If we are in the Lamarckian scenario, what happens is that the mutations arise when we impose the challenge. Okay? So how many cells do we expect to have? How many resistant cells do we expect to have? We have n cells at the end. Okay. How many? And then we put them in this medium where they can grow with phages. So the question is, how many cells will be resistant? I don't want a number like 10. Let's say that you have a quantity that you don't know, which is the mutation rate or the mutation probability. So you know that these cells will have some probability to acquire a mutation. Now we are in the Lamarckian scenario, so these mutations develop. Well, it is not what we think, right? What we think is that these mutations in the Lamarckian scenario develop here, right? So some cells will turn green. Okay. So let's say that we have a mutation probability mu. Mu is a mutation probability. So what do you think is the expected number of mutant cells that we have?
N times the probability.
N times the probability. So we expect that, let's say that the mutant cells are M. What we expect is that this M will be mu times N. Now this is what we expect. What does it mean that we expect is that there is some randomness, right? So this is the average, the expected number of mutant cells. Okay. Now this is the average. What is the distribution? It's a good time for me to drink more coffee because I know that I can wait a bit. So you have n cells, and each of them have a mutation probability. Right? These cells are independent. What is the probability to observe n cells? You have it. Yeah, you were saying something, no? No. So suppose that, I mean, you can imagine that this process is something where you have these cells, and for each cells you can toss a coin, right? And then with probability mu, this cell is resistance. With probability 1 minus mu is not. Okay? Suppose that you have n of these coins and you toss them. What is the probability to observe m heads? This is the probability of observing, if you toss one coin, that's the probability to observe a resistant cell. Okay? But you have n cells. So, who ever heard of a Bernoulli of a binomial distribution? Okay. So this distribution is called binomial distribution. So we can briefly derive it. So what we want is the probability that there are m cells given that we have n cells at the end. Okay? And the mutation probability mu. So imagine that you have n cells, and you say for each of n cells, I can make one of these, each of them resistant. Okay? So this can become resistant, this doesn't, this is resistant, and this don't. Okay? So the process of going from here to here, it's with probability mu. The process of not being resistant is 1 minus mu, and so on. Okay? So we say if we have 1, it's mu. Okay? Now, let's say, what is the probability that I have, that if I have these n coins, I have exactly this outcome? Okay? These are all independent events. Right? Huh? But these are independent events, right? N independent events. So the probability of the full set of events is the product of the individual probabilities, right? So what is the first probability? Mu. This was a test to see if you were alive. You are alive. At least you are alive. Okay. This is mu. What is the second one? 1 minus mu. What is the third one? What is the fourth one? What is the fifth one? Okay? So the probability of exactly this event is mu times mu times mu times 1 minus mu, 1 minus mu. Okay? So is mu to the third, y minus mu to the second. Okay? So is how many times I see the green one, 1 minus mu the times I don't see the green. Okay? So written in this setting here, this term is mu to the m, 1 minus mu to the n minus n. Okay? Now, is this the full probability, or am I missing something? We are missing something because I left a space. So, what am I missing? What I care about is the combinations, right? Because what I care about is not that I have this particular sequence. Any sequence, so also this sequence, would be a good sequence, would be a sequence with an outcome of 3 newtons. Right? Now, so what I have to count is how many sequences there are where I have m, so across all the possible choices of m green out of n cells at the beginning, I have. Okay? And so this number is it's called the binomial coefficient, and it's n factorial. You know this symbol n factorial? Who doesn't know this symbol? It's okay if you don't know. Okay. So it's n times n minus 1, n minus 2, n minus 3, n minus 4, etc. Divided by n minus n factorial, n factorial. Okay. And now, so this distribution, sometimes you see it written as, okay. So this distribution is called the binomial distribution. And it looks like something, so if you have, I don't know, you have n, you have the average, which is n mu, so it's n, and it looks like something like this. So it's something very well behaved. Okay? If m and n are both very large, it looks like a Gaussian. It actually tends to a Gaussian. One other distribution that you might have heard, who have heard of the Poisson distribution? Raise your hand. More people, okay. So this distribution becomes a Poisson if n is very large, mu is very small, okay? So if the number of cells is very large, if the mutation rate is very small, this becomes a Poisson distribution, which is P of n and mu, and this is equal to n mu to the m e to the minus n mu over n factorial. Okay? And for what you're interested in the distribution that is looks like this. Okay? So, what we discover is that in the Lamarckian scenario, what we expect to do is that we expect to have m cells, where m is the mutation rate times the population size. And the distribution is quite well behaved around the mean. Okay? It's approximately a Gaussian, or if you have few mutations, it's a Poisson. So just to say the results here. So if we are in the Lamarckian scenario, thank you. So Lamarck, let's say that P of m given n mu is m mu, n mu to the n, the n e minus mu n over n factorial. And what we care about this is that the average m is equal to mu times n. Okay. And then you can say, okay, this is about the average. Now, if I do one experiment, I will not get exactly the average, right? I will have some variation. So I have to repeat the experiment multiple times. So I start with my bacteria, I do the experiment, and I get one n. I start again with my bacteria, I repeat my experiment, and I get another n. Okay? If you do this, you expect to have a distribution, which is a Poisson with this mean. Okay? And one property of the Poisson distribution is that if you look at the variance, which Javi introduced yesterday, the variance is equal to also to mu n. Okay? This is a property of the Poisson distribution, which means that if you want a measure of the width of your distribution, what you can do is you can calculate what is m squared minus n, so the variance divided by the mean squared square root. Okay, this is called the standard error, and this quantity should be 1 over square root of n. Okay, so this is basically the prediction here. I erased my space. Is that in this case here, when you do the experiment, you have on average n new cells, and sort of the error that you make, what is the how much you can depart from this mean is standard error is 1 over the square root of the average. Okay? Questions about this? If something is unclear, let me know. So this means basically, if you have these things about the standard error, means that if you have a distribution, a Poisson distribution that is centered around 10, more or less this distribution has a width which is between 7 and 13. Okay. If you have a distribution that is centered about 100, okay, the width of this distribution is about 90 and 110. Okay. Good. Okay. So this is Lamarck. Now, let's think about what we expect under a Darwinian scenario. So let's think again about the experiment. The experiment, we start with one cell. This population grows exponentially, arriving to n cells. Okay, and then we put the challenge. And then we count resistant cells. Okay. But now the question is, where do the resistant cells come from? Where the mutation occurs? So in the Lamarckian scenario, they were occurring here. Where do they occur in the Darwinian scenario? Before. So they have to occur somewhere here. Now, where do they occur? In any point. So they can occur, for instance, here. Right? But now, if you have a mutation event here, okay, what happens is that this population, these cells, is one cell that is growing exponentially, is also growing. So all the cells that descend from it will be mutated, right? But then if a mutation occurs here, also all these cells will have a mutation. Okay? So when I put it on a challenge, all these cells will be mutated, all these cells will be mutated. Okay? So then if I take these things and I count how many mutated cells I have, I will have that all the cells here are mutated. Okay? Now the order here, it doesn't mean anything. Okay? It's just looking at the cells sort of in a simple space. Okay? So if you think about this process, there is something we can sort of guess immediately. What we can guess immediately is that it's very different if a mutation arises here. Okay. Because at the end we are going to have few cells. Right? But if the mutation arises here, we basically will have all the cells to be resistant. Okay? So something that we can guess qualitatively, before doing any calculation, intuitively, okay, is that in this kind of experiment, under this scenario, what we expect is that we expect the distribution to be much broader. Right? Because there are experiments where by chance the mutation occurred at the beginning. And if the mutations occur at the beginning, then we expect at the end to have many, many resistant cells in that experiment. If then you repeat the experiment, if by chance the mutation occurs at the end, then you're going to have fewer resistant cells. Okay? So you expect the distribution to be much broader. Okay? Is this clear? This is, in some sense, the most important thing. The rest is trying to get a more mathematical grounding of this, but it's important to have the intuition. Okay? Good. Okay, so let's try to write some equation to arrive to this in a more formal way. Okay. So now this process is more complicated than before. So let's start with a simplification. Okay. Let's start with, for a second, not considering stochasticity, right? Not considering the fact that mutations are a sort of random process. Okay? Let's try to write simple equations that describe the expected frequency of these mutations. Okay? This will not get to us to the distribution, but it's important to sort of shape the intuition. Okay? So let's say that we want to write the equation for the sensitive cells and the mutated cells at time t. So what matters here is, over time, while these cells are growing exponentially, okay, how many cells of each type I have, right? So what is the S dt? So how does the S, so let's first focus. We have n total number of cells. How does this change over time? So So what do I have to write here? This is the dt.
It would be a constant or multiplied by n.
Yes, it will be a constant that we can call r, the growth rate, times n. Okay. And now what we are assuming is that the mutant and the sensitive cells grow at the same rate. Okay. So what is the growth of the sensitive cells? They grow as these ones. So what should I write here? Speak a little bit louder.
A constant times n.
Which constant?
The same one.
The same r. Okay. I should write r times s. Okay. Am I missing something? Yes. What am I missing? The probability of the probability of a mutation. Okay. So what should I write here? So this is the growth rate times s. But they grow at this rate. This should be multiplied by the probability
Of mutation.
of not having a mutation.
Okay.
Right? Because when there is a mutation, I create a new mutant cells. So the growth rate of sensitive cells is this. Okay? Good. So what is the growth of the mutated cells? There are two processes that make the number of mutated cells change. One is the mutations, and the other one is their growth. Okay? So let's start from their growth. How do they grow? Exponential. With what rate? R. This is R times 7. Okay? Then we have another term, which is mu times S. Okay? Now, if you sum these two, you can see that the sum of S plus M is equal, which is ds dt plus dm t is equal to dn dt as it should be. Sorry, is equal to dn dt is also equal to Rn as it should be. Right? All these quantities are the same.
Sorry, I just didn't understand why the mutant cells depend on the sensitive cells.
Because there is a probability that when a cell duplicates, when a sensitive cells duplicate, when it duplicates, create a mutant cells. And so this is this term. Okay. Other questions? Good. Okay. So how many cells, how many mutant cells do we expect at the end? Now to do this, well, in principle we can solve these equations, but there is something a little bit smart that we can do. Okay. And let me think if I want to do that smart thing or not. No, I don't want to do it. So we can solve this equation. Okay. If we solve this equation, what we get is that we get S of t is equal to S of 0, okay, which we can assume being equal to 0, equal to 1. So we have 1 initial cell, e to the r 1 minus mu t. Okay? And then we have m of t, which is equal to the number of cells. In principle, I can solve this equation, but you can immediately derive it saying that this is nt minus s of t. Okay? And so n of t, again, we start from one cell, so it's this quantity. So m of t is equal to e rt minus e r 1 minus mu. Okay? And so what we get here is that this quantity is e to the rt 1 minus e to the minus mu. Okay. And so if we ask what happens at the end of this growth, let's say that it lasts a time t, okay, how many cells do we expect? Well, what we have is that we have the S, n of capital T, so the number of cells at the end is equal to this capital N. Okay? And we have that M, and is also equal to E to the RT. Right? So then we have that M, the number of mutants at the end, which is capital M, is equal to E to the RT, which is N times 1 E to the minus mu. Okay? Sorry, here I was missing an R. Here there is an R, here there is an R. Okay? And if you substitute, you get that M is equal, if you solve for this equation for, well, I already solved. Yes, no. Okay. So you get this expression here, okay? Which depends on N and the indication of N. Okay? So what you get is that as before, you get that N is proportional to the growth rate, and now you have this term here. Okay? Now, if this term, if mu is much smaller than 1, this term can be written as mu RT. Okay? And so what you get here is mu N times RT, but this RT is equal to log N. So this is mu N log N. Okay? Again, this is all written up, so you can follow this. So what we discover is that we have the number of mutations, and at the end, it's something that is proportional to the growth rate, proportional to N, with some small correction, log N. Okay? Now, this is sort of an average in a process that is fully deterministic. Now, what we want to calculate is the distribution. Right? We have this intuition that the distribution will be quite different. Okay? So let's try to calculate this distribution. So let's write somewhere here, well, let's write somewhere here that N is equal to mu N log N. Okay? But let's try to write a distribution. Now, to write the distribution, actually, writing the distribution is not a simple problem, right? Exactly. So we do some intuition, which works very well. Okay. So let's try to write what equation we will need to solve. So let's say that we are imagining that we have our cells, right? And we go from 1 cell to n cells. Okay? So we have the first cell, the second cell, the third cell, the fourth cell, the fifth cells, etc. Okay? And now we can imagine those stacking like continuous line, all the cells based on the order where they are born. We don't care about the time. We just look at the cells in the order they are born. Okay. And then what we ask is that we have n is 1, 2, 3, 4, 5, 6, 7, and so on. Okay. And what? Sorry, small n. just to use the notation. And then when a new cell is born, right, these cells can be a mutated cell. Okay? So this will be 1, this will be 2, etc. Okay? Is this clear, the setting? Any question? So what we have is that we want to write what is the probability of having n cells, in this case 2 cells, 2 resistant cells, when we have n cells. Okay? So what is the probability of having m cells? Sorry. What I want to do is actually to write it as what is the probability of having m plus 1 when I given n minus 1? Okay? So what I want to know is what is the probability that at the next step I'm gonna add a cell that is a mutant. Okay? So this is telling me what is the probability that I have n plus 1, n plus 1. Okay? Now, what are the possible events? What I can write it as is as a combination of two things. Right? So the probability that I have n plus 1 given n plus 1, I can write it as the probability that I have m when I have n, okay, and then the new cell that is born, okay, the new cell that is born is a mutant. Okay, so let's say that this term is, let's write it as a small p, mutant, and n. Okay, so this quantity here, so this is the probability that when a new cell is born, this new cell is a mutant. Okay, times the probability of having n cells. Then there is another way in which I can arrive to this point. Okay? What should I write here? So in order to have n plus 1 mutant given n plus 1 total number of cells, one way is that when I have n cells, I have n mutant, and then a mutant was born. Or
Death.
there is no death. That they already had n plus 1 mutant, and then a sensitive cell was born. Okay? So we look at an example. So what is the probability that I go yes, that at the third step I have, so what are the ways in which I can have n plus 1 mutant, n plus 1, right? So I could have it going from, for instance, this case to this case. So this is a case where a mutant is born, so it's this situation here. Or I can go from this one to this one, where the mutant was already there, and I add one cell. Okay? This is the idea of the two scenarios. Now, these are the only two events that can occur also, right? So when a new cell is born, it's either a mutant or a sensitive. So what we have is that, use the right one. We have that the probability of a mutant being born is equal to 1 minus the probability of a sensitive being born, right? Because these are two exclusive events. When a new cell is born, it's either a mutant or a cell. Okay? Now, what are these probabilities here? So let's think about the probability that a sensitive cell is born. Okay? The probability that a sensitive cell is born is proportional, let's say, to to what? Well, it's proportional to 1 minus mu times S. Okay? So it's the number of sensitivities that there are. So it's like to say, I have n cells, I pick one cell at random and that reproduces. Okay? This is what we're saying. If I pick one cell at random, the probability to picking a sensitive cell is proportional to how many sensitive cells I have. But then if a sensitive reproduces, the probability that a sensitive cell produce a sensitive cell is proportional to 1 minus the mutation rate. Okay? And so this is the term, and this should be divided by n, because I can pick n cells. Okay? So this is the probability that sensitive cells reproduces. Okay? Now this one, if you want to write it in terms of n and n, I can write it as 1 minus mu, and S is n minus m over m. Okay? And so this one is 1 minus mu, 1 minus m over m. And then from this expression I can get this, and then I have this equation that I cannot solve. Okay? So since I have 30 minutes and I cannot solve an equation, what do I do?
You have to check it.
Huh?
You have to check it.
That's a good point, but this is not what we're going to do. Okay? So this is the full process. Okay? And if you are, in the notes there are links to papers that solve this exactly. It's complicated. But if you want to get some heuristics, you can focus on one simple limit. Okay? And the simple limit you can focus on is the limit where mutations are very rare. Okay? So if mutations are very rare, okay, and we focus on this process, what we can imagine is that in the whole process, there is at most one mutation that arrives. Okay? So what I can imagine is that if there is a mutation, there is one mutation event. Okay? So I'm not in a situation where there is a cell mutating here, a cell mutating here, a cell mutating here, a cell mutating here. Okay? What I'm saying is mutations are rare. So there is at most one mutation event. Okay? This is the case I'm gonna put in. Now, you can also appreciate that if mutations are rare, okay, it's not really a problem if there are two mutations happening. Okay? Because if they are rare, one mutation appears here, and one mutation might appear there. Okay? But the mutation there is going to produce a very small number of mutants. So the error I make is very small. Okay? So let's say that we focus on the case where we know that there is at most one mutation appearing. Okay? So if there is at most one mutation appearing, what we can ask is basically what matters here. Let's say that there is one mutation appearing. There is events when the mutation doesn't appear and those are zeros. Okay? If the mutation appears, there is only one thing that I care of. It's when does the mutation appear? Does it appear early or does it appear late? Okay? And so the point is, what I can write is what is the probability that a mutation has not appeared yet? Okay? So let's ask, what is the probability that a mutation has not appeared yet using this equation? Okay? So what is the probability that I have 0 mutant when I have n individuals in my population? I can substitute in this equation. Okay? So what I need to calculate from this is PSMN for m equal to 0. Okay? If I substitute m equal to 0, I get 1 minus mu. Okay? So this is the probability that if I have n individuals that are all sensitive, what is the probability that the n plus 1 is going to be a mutant? It's proportional to the probability that one individual mutates. And then p mu 0 n is simply mu. I know it's simply sorry. Good. So in this case, if m is equal to 0, of course I don't have this term here, right? Because this term is the case where a mutant appears, but there is no mutant. So this term here is equal simply to this term, which is yes, which is 1 minus mu p naught n. Okay? This can be understood intuitively, right? The probability of having 0 mutant when I have n plus 1 cells is the probability that whatever cells reproduces does not mutate. Okay? So now I can solve this equation. I can solve this equation because then I get that this is equal to 1 minus mu, and then this term here I can use this equation, and I get 1 minus mu p0 n minus 1. And so if you solve this, what you get is, if you keep doing this, you get that p0 n is equal to 1 minus mu to the n p0 1, which is equal to 1. Okay? So this is the probability that there were 0 mutant at the first cell, and I assume that that's true. Okay? So basically what we have is that this term is 1. We have that the probability that this is also intuitive. The probability of having 0 mutant when I have n cells is the probability that no mutation occurred in any of these processes. Okay? So this is the probability that I have no mutations at the step n. Now, if I want to know what is the probability that the mutation occurred at the point n mu, how do I calculate this? Do you know it? So this is the probability that no mutation occurred at the time at the population n. And I want to calculate what is the probability that the first mutation occurred exactly when I had any individual. No. I have to calculate from this. So this is telling me what is the probability. I'm sort of having this process where I check if a mutant appeared or not. Right? And this probability is decreasing as the population is growing. Now, the probability that a mutation occurred exactly at the point n is equal to the probability that I have 0 mutant. The probability that I have 0 mutant at n, okay, minus the probability that I have 0 mutant at n plus 1. Because the difference between the probability that, what is the difference between the probability that there were no mutant at n and no mutant at n plus 1 is the probability that there was one event. Okay? And so if you substitute this expression, what you get is you get 1 minus mu minus to the n. 1 minus mu to the n mu plus 1, which is equal to 1 minus mu to the n times mu. Okay? So this is the probability that the first mutation events occurred when I have exactly n individuals. This is n mu equals 2. Okay? And so my assumption is that I have the first mutant, and then that's it. The population will grow exponentially of the mutant. So, yes.
I am followed by the second term there. We were given n plus 1, but then we also we have n, and then 1 was born. And that was also yes. But
That's true. Sorry.
I guess it was, but we were given n plus 1, but then we were assuming we were not given n plus 1, but we were given n.
Yes, we are looking at the previous step. Right? We are saying what is the probability that I have n plus 1 mutant at the next step. So,
More like a jump probability.
no, this is a conditional probability. Suppose that I am at the step n plus 1. What is the probability of having n plus 1 mutant? This term is the probability that I had n plus 1 mutant before, and then a sensitive cell was born. And this is the probability that I have m and the mutant was born. Sorry, this was a mistake. Okay, but it doesn't affect.
I think there the third line was right. I mean,
No, it's okay because this is 0,
Yeah.
so it's okay. Okay. So what I have is that what we're saying is there is the randomness come from this n mu, which is the number of individuals where the first mutation appeared. And so this n mu is some random variable with a distribution which is mu 1 minus mu to the n. Okay? And then I'm saying where this mutation appears, dm dt will grow how? Will grow exponential. Will grow as r times n. Okay? We're almost there. And then we have also dn dt equal to r n. Okay? Right? So a nice thing I can do is that I can write dm dn. Okay? Which should be equal to m over n. Okay? I know. And then I can solve this by part, right? So actually I was preparing this part of the lecture on the plane, so I didn't have internet connection. So I spent like 3 hours saying, shit, what is that? Like sort of kicking the seat in front of me. I succeed. Okay? So I can solve this and integrate, right? Now, what I want the last input from you is where are the limit of the integration here? So m will go from 1 from the first cell, right, to the final number of cells. N will arrive to capital N, but where it will start from? So this is the value of the population where the first mutant arrived. N mu. Right? So I'll have 1 mutant cells when the population is N mu. Okay? So if I integrate this, what I get is, well, this becomes log m, log 1 is 0, so I get log m is equal to log n over m. Okay? So what I discovered is that m is equal to n over n mu. Okay? What this means is that the fraction of cells that are mutant is conserved. Since both are growing exponentially, whatever was the initial fraction of mutant cells is conserved to grow. And now what is n mu? What is n mu? It's a variable. Right? But we know distribution. And so we know the distribution, and distribution is this one, right? So I can derive the distribution of m from the distribution of n mu. Right? So suppose that I have a distribution p of x, okay? And I have some function y of x. How do I get the distribution of y? Do you know this? There is one thing, which is, you can imagine, I mean, I don't know if you will explain this. You can, but no.
I don't know.
So the trick is that what is conserved, so let's get an intuition for this. So the probability that I observe, if I have, I don't know, if I'm asking what is the probability that what is the probability that I'm gonna win? I mean, I play a game, I play a lottery, and I win some dollars. Okay, I can get what is the probability that I win the same amount in Quetzal. Okay, and of course, not the same amount, meaning $10 and Quetzal, but like with the fraction. Right? And so if I look at any possible event, right, simply this event corresponds to the transformation value. So this is expressing this thing here, right? I have a way to transform that, and whatever interval of events I'm looking at, let's say the probability that I win from $10 to $100 corresponds to the probability of winning in that interval. Okay? And so this means that if I want to have the probability of y, what I need to do is to calculate dx dy times p of x of 1. Okay? Okay. If you lost me this, there is a way to do this change of value. Okay? And the way this works is that here we have that the probability of n given n and mu, okay, will be equal to but from this one I can get n mu equal to n over m. Okay, so this will be the probability of n mu. So where in this function I substitute this expression times the derivative of n mu dm. Okay, from which I get that p of n and mu is equal, I have to calculate this derivative, so that is like an absolute value, and here I get n m over 2, and then I substitute it here, and I have mu 1 minus mu to the n over n. Okay? So I get an expression for this distribution. Now, this is a moment to stop. So before I stop, questions. Okay. So what we're gonna do, I don't know when, this afternoon, I guess, is that this is the end of the calculation. We're gonna look at this distribution. Okay. The key point here, the key point I want you to remember is to have this intuition that what makes the situation here different is the fact that mutation can arise early or late. And since the population is growing exponentially, if it's arising early, this quantity will be very large. If it arrives late, this quantity will be very small. Okay? And so we have a Y distribution. Okay? This is the Y distribution, and we're going to look at it later. Okay?